Today I'll be discussing APOE and its role in the development of Alzheimer's disease. Our learning objectives are to explain the basic role of APOE in normal CNS function, we'll identify some mechanisms responsible for the role of APOE4 in development of Alzheimer's, and we'll examine the differences in Alzheimer's presentation within patients who carry APOE4 versus patients who don't. For an overview of Alzheimer's, it's a neurodegenerative disease primarily characterized by progressive loss of memory and other cognitive functions. The hallmark of this disease includes a buildup of amyloid plaques and neurofibrillary tangles, which will eventually lead to the death of neurons. And the severe degeneration seen in the brain typically begins in the medial temporal lobe, which includes the hippocampus and entorhinal cortex. And as the disease progresses, we see it spread to structures such as the neocortex, which includes the temporal and parietal lobes, the frontal lobe, and in later stages, the occipital lobe. APOE is the gene responsible for the creation of apolipoprotein E, which is the most abundant apolipoprotein in the brain. Its primary role is to transport cholesterol and other lipids to neurons, where these lipids play a role in synaptic function and injury repair. In the brain, APOE is mainly produced by astrocytes, but we do see it expressed by other glial cells, such as microglia. And over here, we see the general structure of a lipoprotein. We see the phospholipid layer, which encases the hydrophobic cholesterol and triglycerides. And over here, we see the apolipoprotein, which wraps around the phospholipid layer and gives structure to the lipoprotein as a whole, allowing it to move within the aqueous environment of the brain and body. The predominant APOE alleles in humans are APOE2, APOE3, and APOE4. Each have a different impact on the risk for developing Alzheimer's disease, with APOE2 reducing that risk, APOE3 carrying a neutral risk, and APOE4 being a major genetic risk factor for developing Alzheimer's. While the frequency of these alleles will vary by ethnicity, in general, APOE3 is the most common, followed by APOE4, and then APOE2, which is the rarest allele. Each allele codes for a slightly different isomer of APOE, which results in the differing impacts that each have on Alzheimer's disease risk. APOE is a sequence of 299 amino acids, with the difference between each isomer being at just 2 positions on that whole amino acid sequence, specifically position 112 and position 158. APOE4 is implicated in several important areas of Alzheimer's pathology, which include the development of beta amyloid plaques and neurofibrillary tangles. We see its promotion of inflammation in the brain, dysregulation of lipid metabolism, and a breakdown of the blood-brain barrier, each of which will be discussed in the subsequent slides. APOE4 promotes plaque formation in several ways, including by accelerating early seeding of beta amyloid. So in Alzheimer's patients who carry APOE4, we see plaque formation in earlier stages of the disease versus patients who do not carry APOE4. It also enhances fibrillogenesis, which is the aggregation of beta amyloid fragments into beta amyloid plaques. And we also see a reduction in the ability to clear beta amyloid as it begins to build up in the brain. And this will be further discussed in the following slide. APOE4 also enhances deposition and negative effects of neurofibrillary tangles. It does this by increasing tau phosphorylation, which is the increase in the amount of tangles within the neuron, and it exacerbates tau-mediated neurodegeneration, which is essentially the negative effects that the tangles have on the neuron that eventually lead to cell death. The role of APOE4 in neuroinflammation is currently still being researched, so there are many mechanisms that aren't fully understood just yet. But currently, we do know that APOE appears to modulate neuroinflammation by influencing the microglial response to stress. In this case, that stressor is beta amyloid aggregation. And the effectiveness of this response appears to be isomer dependent. APOE4 appears to be correlated with a negative outcome or negative response versus APOE2 and APOE3. APOE4 seems to promote inflammation both directly and indirectly. We already discussed that there's a reduced ability to clear beta amyloid. This may be due to a less efficient microglial response, which results in further beta amyloid induced inflammation downstream of this inefficiency. Essentially, microglia associated with APOE4 are less likely to migrate and then subsequently dispose of amyloid plaques as they begin to build up. APOE4 is also associated with overreactive microglia, which overproduce inflammatory cytokines in response to routine stimuli, and this is an effect observed even in patients who do not have Alzheimer's disease. So this does raise questions about APOE4 and its cognitive risks independent of Alzheimer's disease, which will be explored later in this presentation. A hypothesis for how this pathway works is that impaired APOE4 receptor signaling may lead to beta amyloid-induced activation of toll-like receptor 4, which is a pro-inflammatory pathway, and suppression of interleukin receptor 4, which is an anti-inflammatory pathway. It's thought that APOE4, since it's known to be less lipidated and less abundant in the brain, that this leads to reduced APOE receptor binding and agonist activity, which allows for beta amyloid to promote pro-inflammatory signaling and suppress anti-inflammatory signaling, leading to a negative neuroinflammatory phenotype and ultimately neuronal dysfunction. Inflammation is also going to be discussed in the following slide as we go over lipid metabolism. So dysregulation of lipid metabolism appears to be upstream of neuroinflammation. This is due to altered APOE4 induced lipid metabolism manifesting as the accumulation of lipid droplets within microglia. This accumulation causes phenotypic change in these cells, which is characterized by reduced phagocytic ability and increased release of pro-inflammatory factors. This may explain the semi-paradoxical points that we made in this slide, which discuss the less efficient microglial response, while the microglia are also overreactive and pro-inflammatory. This appears due to this phenotypic change. This accumulation appears to be related to APOE4's deficiency in facilitating the efflux of cholesterol, which is potentially linked to APOE4 carriers having lower levels of overall APOE. So essentially, because there's less APOE in the brain, it is less efficient at removing cholesterol, causing a accumulation and then subsequent deposition into microglia, which once these lipid droplets accumulate in microglia, this marks its transition into this phenotypic transition into what's called a disease-associated microglia, which are characterized by increased inflammatory signaling and a decreased beta amyloid clearance ability. Research is coming out on APOE, on the general role of APOE in maintenance of the blood-brain barrier. Emerging evidence indicates that breakdown of the blood-brain barrier may be one of the earliest pathogenic events in Alzheimer's progression and is also potentially implicated in general cognitive decline independent of Alzheimer's. APOE4 specifically is associated with decreased proliferation of endothelial cells and a degeneration of parasites, which is thought to ultimately lead to the breakdown of the blood-brain barrier. In APOE4 carriers, this breakdown contributes to cognitive decline independent of beta amyloid and tau pathology. So again, there seems to be some link between the importance of APOE in maintaining the blood-brain barrier and the deficiencies of APOE4 leading to cognitive decline independent of the other factors that we discussed that are involved in Alzheimer's pathology. And as a note, this breakdown appears to begin in the medial temporal lobe, which will be important in a second when we go over APOE4 carriers versus non-carriers. So while we know that APOE4 is definitely a risk factor for developing Alzheimer's. It's unclear if APOE4 causes more rapid cognitive decline versus what we see in non-carriers. The general consensus currently is that carriers and non-carriers seem to follow a similar rate of decline. The differences arise in the cognitive presentation, with carriers suffering greater memory deficits than non-carriers, and non-carriers suffering greater non-memory deficits such as in executive functioning. These deficits appear to correlate with differences in volume loss in certain areas of the brain. Carriers show greater atrophy in their medial temporal lobe, so we see here degeneration of the hippocampus and entorhinal cortex, which may correlate with the more pronounced deficits in memory that this group experiences, while non-carriers show greater atrophy in their frontoparietal lobes. So again, here we see degeneration in the frontal lobe, which may correlate with the more pronounced deficits in executive functioning that this group experiences. Overall, these differing effects demonstrate the variety of complex mechanisms involved in Alzheimer's and elucidate the need for research beyond the current hypotheses that we have. Alzheimer's research has largely revolved around the focus on amyloid and tau pathology, but as we've seen today, APOE4 presents in several under-researched areas, some of which are independent of classical Alzheimer's pathology, and may also play a role in cognitive decline independent of that pathology. This presents an opportunity for research into fields such as isomer-specific targeted therapies, which would include modulation of ApoE levels and ApoE lipidation, allowing for therapy to be tailored specifically to someone's genotype. Further research is also needed in promoting cerebrovascular integrity, which, as more research has been coming out, demonstrates that this integrity plays a crucial role in staving off cognitive decline, again, independent of Alzheimer's disease, but is obviously exacerbated in patients who do suffer from Alzheimer's. So placing more research into this field to understand how we can promote the cerebrovascular and blood-brain barrier integrity is important in the upcoming years. We also need a better understanding of how ApoE modulates inflammation. Currently, what we presented today has mainly been hypotheses. Some mechanisms have been worked out, but for the most part, we don't have a concrete understanding of exactly how it's modulated. So overall, this really illustrates an understanding that something is going on with ApoE, even outside of APOE4, and that it may be a target of therapy in future research opportunities. And these are my references. Thank you.